Okay, I'm going to start in 30 seconds. Oh, I got a text. I should start. Um, so, I don't know what to do with myself. My first time. Thank you to David Barclay and all at Dal who have invited me to give this virtual seminar. Um, yeah, it's been interesting so far, right? These past couple of weeks. So, I'm going to have the comments over here. I think there's a little bit of a delay, so bear with me. Um, okay, so I'm sorry, I had one note. So yeah, today going to be talking about iron fertilization, basically how iron and nitrogen cycle in these high nitrate iron deficient regions. And all this with the focus on, carbon fixation and the biological carbon pump. So, but before I get going, two items on the agenda. First, I'd just like to recognize all of the great work that's come before me. Of course, I stand on the shoulders of giants, and this is particularly true for the work that's been done to understand iron cycling in the ocean, which I'm a relative newcomer to. I just want to drop some names here, people I talked to, but also people, of course, whose work I've looked into. So I'm talking about Barbeau, Brulin, Brzezinski, Boyd, Bundy, my friend Randy Bundy, who's been really great and helpful. Hutchins, Strepek, of course, John Martin, Chris Measures, Karen Self, Julie Granger has been a huge help. My co-authors on the previous paper, Kate Mackey and Danny Sigmund. And then I also had a fantastic conversation with Ben Twining at the last Ocean Sciences meeting, which hopefully will not be the last, right? But it feels like it was a couple of years ago. So the second item, so that's the first item on the agenda. I just wanted to make sure I get that out right away. And I'm just checking my feed to make sure I'm still working correctly. And I guess the second item on the agenda is that I got to turn myself down. Second item on the agenda is just to let you know where I'm coming from. Sometimes I refer to my training as, in marine biogeochemistry as, in the Sarmiento and Gruber School of Marine Biogeochemistry. And, up until a couple of years ago, I thought that was marine biogeochemistry. But it's pretty clear now that there are those who approach marine biogeochemistry with the hammer of geochemistry, and those who approach marine biogeochemistry with the hammer of biology. And it's been really, really exciting and also at the same time challenging to sort of move myself and try to understand more of what the biologists are saying. So I just wanted to get that out front because that's the focus of this talk really, or sorry, the approach to this subject matter is really me coming from it as a geochemist, and in particular, someone who's spent a few years thinking about nitrogen in the ocean, and then crossing over and saying, okay, well now I don't really care about nitrogen that much, right? I care about carbon and carbon cycling in the ocean. So how do I get there? Oh, well, there's unused nitrate in the ocean, iron fertilization, all that. So let's get going. Okay. So these are iron limited regions. This is a plot of surface nitrate concentrations, and I've color coded it just one color. This is everywhere that's greater than 1 micromolar. I've color coded in this lovely pink or whatever color, and that's just to make this very big point that these are iron limited regions, and by that I mean if you add iron to these waters, you increase nitrate consumption, increase phytoplankton growth, therefore increase carbon fixation and ultimately influence and strengthen the biological carbon pump, the export of carbon to the deeper ocean. And so this is, historical stuff going back to Dugdale and Gehring and others. So what I mean by that is just to connect the dots. Nitrate consumption leads to this new primary production, biological carbon pump efficiency. And so you can think of this unused nitrate in the surface ocean as a missed opportunity to sequester carbon in the ocean away from the atmosphere. All right, so this is sort of the motivation. Okay, so now pretty important question that I've been seeking answers to for a few years is, so how do you get variability of nitrate consumption in these regions? And so there's essentially two ways that have been proposed to do this. You could change the ratio of nitrate to iron supply in the ocean. Sorry, that's being upwelled to the surface water. So you change the ratio, you increase how much iron is coming up to the surface relative to nitrate. An alternative to that is a change in the supply of iron-rich dust to the ocean. And so this is John Martin's famous iron hypothesis. And so there's this kind of legendary quote that if you give him a half a tanker of iron, it will give you an ice age. So what he's saying is we're going to take advantage of this missed opportunity to sequester carbon in the ocean by relieving the iron deficiency in these nitrate-rich, iron poor waters. Okay, so I like cartoons. I like drawing pictures of things, different ways to explain what I'm getting at. So this is sort of my picture of the iron hypothesis, or sort of what we think, a different way to think about nitrogen and iron cycling in the ocean and the fixation of carbon. So here we have some surface water mass. We have nitrate and iron being upwelled in some proportion. They are taken up by phytoplankton. There's some contribution from iron-rich dust to the surface, and this somehow works to fix carbon and have the carbon exported from the surface ocean. Okay. However, there are cracks, what I'm calling cracks in the iron hypothesis. And so these are based on paleo-oceanographic data sets. Now, I'm not saying that what I should say is that we know if you add iron to these nitrate-rich, iron-poor waters, you increase nitrate consumption and in most cases increase phytoplankton growth. But looking at over these longer timescales, these ice age timescales where we had these global changes in dustiness, essentially, and presumably increased supply of iron-rich dust to the ocean, which has been shown in through using a couple different proxy records. This is a good way to test this idea. So, okay, sorry for all that background to it. But what we find is by using proxies to reconstruct nitrate consumption in these nitrate-rich, iron-poor regions, both the equatorial Pacific and the Southern Ocean, we find these either no or apparently little influence from iron-rich dust on nitrate consumption, or huge changes in nitrate consumption that occur without any change in atmospheric dustiness. Okay, so that's a big outstanding question right now, is how do we bring these all together? So here I'm going to provide you with sort of a synopsis of where I'm going to take you in today's talk. And so this begins with a proxy record of nitrate consumption in the eastern equatorial Pacific. And so this is what we're showing here. I'm not showing any isotopes because I know isotopes sometimes freak people out. I'm just going to say nitrate consumption is upwards. And so that's, to give you the framework here, the x-axis is the age, in thousands of years. It's 400,000 years towards the present on the right. The top part of the plot is nitrate consumption in red, and below that is dust concentration from Vostok ice core. And so what you see here is that this sediment proxy for nitrate consumption is inconsistent with the iron hypothesis. They don't look alike, right? There might be some overlap, but there's plenty of other variability in nitrate consumption that we are not finding that we're not observing in the nitrate consumption. So, to kind of give you a synopsis of today's talk, it starts with this sediment proxy data, both in the equatorial Pacific and the Southern Ocean, that is inconsistent with the iron hypothesis. What I did in my postdoc was I said, okay, well, let's go out to sea and let's analyze nitrate consumption in these areas. And what I did was I developed a new method for quantifying nitrate consumption in these regions, and using nitrate isotopes. And so using this new method, I was able to quantify how much nitrate consumption is occurring there. We found this huge mismatch between how much nitrate consumption is occurring and how much iron is actually supplied, and how much predicted nitrate consumption you would have there. And so we developed, through this, I developed a new model for elemental cycling in these nitrate-rich, iron-limited waters. The last part of the talk, I will then take this model and use that to reinterpret the sediment proxy data. Okay. Okay. So we're going to look at four iron-limited laboratories. And so 4 regions. The equatorial Pacific is going to be the first one we look at, and then we're going to go to the Southern Ocean and the subarctic, Pacific and Atlantic. And so what I'm going to do is I'm going to use nitrate isotopes to identify the source water nitrate concentration. And then I'm going to take that source water nitrate concentration, calculate how much nitrate consumption there was between that subsurface source and the upper mix layer. And then I'm going to make a couple calculations about, very simple calculations about, well, how much potential consumption should we have based on how much iron is there. One point about identifying the source water nitrate concentration, we can apply that to all the other geochemistry, right? All the other characteristics of the upper ocean that we're interested in. And the idea is to identify the characteristics of the waters just before they enter the euphotic zone. So it's sort of like a T0, or the initial conditions. So we're going to take a look at the eastern equatorial Pacific, and let me take a sip. So before we drop into the eastern equatorial Pacific, a reminder of how, when I'm saying we used to think about the system, I think this is this is how I was taught how these systems worked. And what I was taught was what new nutrients were added to the upper ocean are what matters for carbon fixation, and that's relevant for carbon export to the deep sea. Okay, so I'm looking at this through the lens of nitrate consumption in the ocean. So this is my specialty. Okay, so first problem with trying to identify the subsurface source of nitrate is, I think it should be pretty obvious, right? We are, here's a kind of an example of nitrate concentration with depth in the eastern equatorial Pacific. We see concentration on the x-axis down to 250 meters on the y-axis, and you see this continuous curve. If you were to look at the vertical structure of this water mass, I mean, it does not look continuous. There are subsurface currents going either way, zonal currents. There's no very clear relationship between the thermocline and how much nitrate is there. It's just not easy to identify, what the initial nitrate concentration was of the waters that were upwelled to the surface. You can make some assumptions like, oh, let's just choose the thermocline, or let's choose the first derivative, or let's choose, the equatorial undercurrent, which would be a good choice actually, because that one that appears to be the source. But coming at this without making any assumptions is kind of is challenging. Sort of having some way to base the initial nitrate concentration on something more fundamental is not as easy when you're just looking at nitrate concentrations. So enter nitrate isotopes. So I'm going to let you take this in for a second. So here we have measurements I made at 0 north, 110 west, so right in the heart of the eastern equatorial Pacific, cold tongue. And so this is same station over 5 years, spring and fall. So this is on the schedule of NOAA's ship that tends the El Nino buoys that are out there. Okay, so I've overlapped all of the data here. The colors are, and the symbols all mean something. I guess you could look at the paper if you want to see more details. But the x-axis is nitrate concentrations. The y-axis is nitrate delta N15, so it's the nitrogen isotopic composition of nitrate. This will be the only isotopes you will see in this talk. So please don't run away screaming yet. I'll try to make this as easy as possible to understand. So these are measurements from the upper 200 meters at the same station over 5 years. You could see, all right, first thing we see is they overlap very well. Whatever this pattern is, all the seasons seem to fall on the same trend. And so moving from, I don't know where I am, but moving from your right to your left, you see a change in nitrate concentration, but no change in the isotope, right? So the y-axis is not varying very much. And then there's one point where they all start to change together. We have a lowering of nitrate concentration and an increase in nitrate delta N15. So this is a predictable rise in delta N15 with lowering nitrate concentration as nitrate consumption is moving forward. So this has an isotope effect of 6 per mil. It's all in this paper from 2016. I think the starting point was about 15 micromolar. There's some interesting physical oceanography that this tells us too. But what we're interested in here is that identifying the source waters, right? What's the condition of, or the characteristics of the subsurface waters right before they enter the euphotic zone. So this gives us a way to estimate that in a kind of, I would say, a more robust way. So we're seeing exactly where nitrate consumption is happening. We can mathematically solve this to find this hinge point, or what some people call a kink. So now, all right, so the isotopes are behind you, I think. I think that's all the isotopes, yeah. So if you were worried, there's not going to be any more isotope talk. So what can we do with this? So we know over the course of this time period, which, I could think of a question someone's going to ask me already. So the samples were taken from 2003 to 2007. There were no huge El Nino or La Nina events, but there was seasonal variability. There's always seasonal variability, and there was some sort of moderate, mild, and so variability. Okay, so back to the slide here. So now that we have this understanding of what the initial conditions of the waters were right before they entered the euphotic zone in the eastern equatorial Pacific, and with the understanding that upwelling is essentially vertical here. We don't have a lot of lateral input at the equator because of Ekman divergence, right? So if you're at the equator, you're essentially seeing waters that came from right below. Okay, so to quantify nitrate consumption, we could say, oh, we have this source value. Let's take the difference from that and the upper mix layer, surface mix layer. And so you see this variability. You see a max maximum nitrate consumption, which is in boreal spring, and you see minimum in boreal fall. I should say that boreal spring is weak upwelling, boreal fall is strong upwelling, and we have an average here. So there is this interesting relationship between upwelling and nitrate consumption, and I'm going to call this the first mystery. So why is there any relationship between consumption and upwelling? Okay, so that's kind of take that and put that in your pocket, and we're going to return to that in a second because there's an even kind of more fundamental mystery, and that comes when we calculate the potential nitrate consumption. So to calculate the potential nitrate consumption, we've said, okay, we know the waters are coming from this depth. What's the iron look like there? What's the dissolved iron? What's the particulate iron? Okay, we also know we're at this location, how much dust input, and the solubility of how much iron comes out of that dust, and yada yada yada. So we can make a calculation, right? How much potential nitrate consumption we should expect based on the iron supply to this region. This makes some assumptions, but I'll walk you through it. So the big point here is that the potential nitrate consumption is crazy small. Okay, I have two bars here, these black bars, and you could see that they're like within the margin of error. They're tiny, and this I would say these are using the best assumptions. So measurements of iron, dissolved iron, and particulate iron at that site. Also sort of boosting up how much dust is available. I allowed for 30% of the particulate iron to be biologically available. Okay, so so now we have this second mystery of why the observed nitrate consumption is so much higher than the predicted nitrate consumption. Okay, if you're anything like me, though, you're saying, well, I don't know what went into these calculations. So first, let me walk you through really fast, just some sensitivity tests. You could take a screenshot or whatever. We can talk about it later. But I would say these are in order of increasingly crazy assumptions that went into this very simple calculation. And yet, none of them can replicate the observed range of nitrate consumption. So the mystery still persists. But wait, there's more, because this simple calculation only considers diatom iron requirements. So what I mean by that is that diatoms are by far the dominant consumer of nitrate, I would say globally, but definitely at this site. So what these calculations assumed were that all of the iron is going just to help them consume nitrate and grow. But we know that's not true. Actually, of the phytoplankton biomass, the diatoms only account for about less than 10% of the biomass at this site. So in other words, what I'm saying is that this is overestimating the iron that's available for nitrate consumption. In other words, there's other bugs there. There's other phytoplankton that also have their own iron requirements, and so this assumption is just not reasonable. So it's actually, even though I've made these potential nitrate consumption estimates, these are overestimating how much iron is actually available to fuel this nitrate consumption. Okay, so now we're going to jump ahead to these all four iron limited laboratories. It's going to be pretty quick at this point because I know I'm already getting behind. But, so let's jump. North Pacific, the left, I should point out, so North Pacific, Eastern Equatorial Pacific, Southern Ocean, and North Atlantic. The left side is based on the observations, which are using nitrate isotopes in all of these regions, and then mathematically determining what the subsurface source was, and then taking the difference with the surface mix layer. And then to the right, in the shades of gray, are the sensitivity analysis. And so you can see that for the top three nitrate-rich, iron-deficient regions, we're not matching the variability of the observed variability of nitrate consumption. The North Atlantic is an interesting, different, is a little bit different, right? So we can sort of match the variability, and this makes sense, makes qualitative sense because the Atlantic is known to have more iron supply to it. But remember, once again, that none of these calculations are taking into account the various demands for iron from the phytoplankton communities in these sites. So, so we're looking at a very large deficit in iron. You could think of it as these places, these iron deficient regions, are even more iron deficient than we thought. Okay, so the big question now is, where is the missing iron? Some people have, I forget who, I think Farouk Azam quoted this as the riding the quote Ferris wheel, where it's getting recycled essentially. And so this is my first statement at the top was to recognize all the work that's come before me, and here's just some of the work that has looked into iron cycling in the upper ocean. So we know that in the euphotic zone there is some degree of iron regenerated for biological use. So, for example, in the equatorial and the subantarctic Pacific, we know it's a large part of the surface ocean iron budgets, and also in the subantarctic Pacific. This is Strepek and Boyd's work, and then has also been suspected to fuel diatom blooms. But the kind of the new thing I'm adding here is I'm saying, okay, well, how does that all relate to nitrate consumption, which historically is the link the nutrient that we're interested in when it comes to what they call new primary production, biological carbon pump, and sequestration of carbon in the ocean. Okay, so I try to use the term differential cycling of iron. This was after talking to Ben Twining. I think this is probably a better way to think about it. So like iron and nitrogen are being cycled in the upper ocean, but they're being cycled differentially. And so essentially what I'm calling on is for a faster recycling or differential cycling, faster cycling of iron in the upper ocean relative to nitrogen. So this is a whole bunch of information just to let you know that to take into account these other phytoplankton groups and their iron requirements and iron scavenging, which has come later in my modeling. I built this box model, has all this stuff. Essentially, for the sake of this talk, what you need to know is this differential cycling of iron can work to explain the nitrate consumption. And what I mean by that is that I used the box model to calculate some required rate of iron cycling in these in the eastern equatorial Pacific. I've also applied it to the Southern Ocean and other regions. And I mean, it's reasonable. It has to cycle faster, it has to recycle faster, but the only observations we have from Strepek in the subantarctic, it's like 2 times faster or something for the equatorial Pacific, which is I think is reasonable. So that's kind of nice to find, that you build a model and it doesn't. Well, you build a model to kind of disprove yourself, right? And but we found that it seems to be reasonable. Okay, so let's jump back to our original view of nitrogen and iron cycling in the ocean, and so we have this is focused on what's added, what's new to the upper ocean, and that's what's in this old view. That's what is driving the biological carbon pump efficiency. However, the nitrate isotopes and the calculated nitrate consumption is inconsistent with this, because the new iron, the iron that is added to the system, cannot explain the nitrate consumption. I think I can say that without any doubt. So I propose this new view, and this new view is that sure, we have added nutrients, but the microbial community is differentially cycling iron relative to the other nutrients that are being added to the system, and that this ultimately must be taken into account when we're modeling these systems, and especially when we're trying to consider their influence on the biological carbon pump. Okay. So this was the second mystery. So we have these two mysteries. The second mystery is why the observed nitrate consumption is so much higher than the predicted nitrate consumption. I think that's because we have differential cycling of iron. This allows for more iron to be available to the diatoms and other nitrate consumers, as well as the entire phytoplankton community. But we have this remaining mystery, this first one I'm calling the first mystery. Why is there a relationship between consumption and upwelling? Okay, so to remind you, because I don't think it's in the next slide, to remind you, when we have reduced upwelling, we have increased nitrate consumption. Okay, let's keep that in mind. So I think we can explain this by linking everything with physics. I think we have to. And some people I know say everything starts with physics. So let's just start with this model upwelling zone. Here's another cartoon for you. So you have some euphotic zone with a fixed depth, and you have below that the source water, the initial conditions, the T0, whatever you want to call it. Now imagine we have a change in upwelling. Now, if we have weak upwelling, you can imagine that across that fixed depth that we would have a longer residence time of those waters that are being upwelled through the euphotic zone. Now, this contrasts with time periods when you have stronger upwelling, when you would have a reduced residence time of those waters in the euphotic zone. And what I'm proposing is that this plays out like this. If we have stronger upwelling, which in the Eastern Equatorial Pacific is in the boreal fall, we have its reduction in residence time. This allows for less iron to be recycled, and therefore we have less nitrate consumption in the Eastern Equatorial Pacific at that time. So I think this is, I was so excited to kind of develop this and come up with this, because it links ocean physics with biology and chemistry, and I think is just offers some opportunities to sort of think about these systems in more holistically, and then ask new questions as well. So bring back to the beginning, to make the beginning begin again. Remember, I had this synopsis where I started with the proxy results, and then I said I had to go out to sea, develop these methods, propose a model, and then use that model to reinterpret the proxy data. And so that's where we are now. We're reinterpreting the sediment proxy data. Okay, so revisiting these Eastern Equatorial Pacific results, I have nitrate consumption. Remember, this is 400,000 years now, right? So I know this might be a little bit of a brain twister, but looking at all these different timescales. So over the last 400,000 years, we have this proxy record of nitrate consumption in red, with a what we call high frequency variability, about 19 to 23,000 years variability compared to the dust concentration from the Vostok ice core. And so the question was, how do we get this variability in nitrate consumption that's seemingly independent of the dust supply? And I'm arguing that it's physics. And this is a little bit of tooting my own horn, but I was probably right when I proposed in this paper way back in 2012 that changes in upwelling are linked to nitrate consumption and ultimately are driven by seasonal heating. And what I mean by seasonal heating is changes in the Earth's precession. And so it changes the seasonality. And I can show you that really quick. So here we have nitrate consumption, local insolation, so 0 north insolation, and then dust concentration. And so you could just see, I've done spectral analysis of course, but this is more fun than spectral analysis. You can see how well this fits. So the driver, I argued before, I didn't know have a mechanism, but I argued that it really looks like insolation is driving upwelling, and somehow this is linked to nitrate consumption. And now I have this model to support it. Okay. And so if I look at my clock, I'm pretty much out of time, so I'll run through really quick just another application. This is work that's in prep to the Southern Ocean. First, I can show you here, this is the past 220,000 years. The CO2, the ice core CO2 is up top. This is a record of nitrate consumption for the most part. We may have some questions about that. I see Pierce is looking at the video, and this is exactly what I was talking about, Pierce. But okay, let's just assume it's nitrate consumption. It's probably mostly changes in nitrate consumption with increased consumption upward. I mean, without knowing what the red proxy record is, it should just be visually appealing, right? Like, look at this mirroring between the atmospheric CO2 and this marine proxy record. It's pretty wild. And so we've been wanting to explain this, but it's challenging to explain because here's the dust concentration. And so you can see the dust concentration doesn't look like the nitrate consumption all the way through. And in particular, we see this time period, which is the onset of the last ice age, where CO2 just starts to dive down, where there's no change in atmospheric dust, and you would presume no change in iron fertilization of these nitrate-rich waters, and yet there's this large climb in nitrate consumption. So my argument is that this is probably increased stratification of the Southern Ocean surface. And so one way you can think about it is we start falling into an ice age through stratification of the Southern Ocean, and that's being shown here. So it's kind of a cool insight to how global climate varies on its own. Okay, so I'll skip through that stuff, and just to summarize, you know, it seems like this differential iron cycling drives most of this new primary production, so most of the nitrate-fueled primary production. Probably, and in my box model, it fuels most primary production, period. You know, it's necessary to explain the growth and biomass of all the phytoplankton in these nitrate-rich iron-limited waters. Then I propose this, so I have this model for differential cycling, and I link it to upwelling rate and have this sort of modulation of iron cycling and therefore nitrate consumption. I think this helps explain a couple mysteries in the paleoceanographic literature. So I am done here. I've never given a talk like this, so it's weird to not have any feedback except comments that I'm not sure are being updated. So feel free to email me or check out my website or hit me up on Twitter. It says I'm not unable to chat, but I guess I'm done. I don't know how to know if I'm done or if I'll get any questions. I'll go back to an interesting screen. That's interesting. Maybe I'll go to something. So first comment was to tell you about my Wes Anderson color palettes. I can tell you to go to prafter.com and you can download them. They are made for Ocean Data View, and so they're a lot prettier, I think, than the available palettes. And when I made them, apparently I didn't realize somebody had already done it for R and MATLAB, but I hadn't known that. A science question: does your box model use the N15 and the delta O18 to estimate nitrate consumption? This is from Pierce. Yes, it does. I didn't bring up delta O18 in this talk because people tend to get upset about isotopes. But yeah, so it's just an additional factor. And if you really want to get nerdy, the N15 and O18 tell you essentially the same source water characteristics. And so that's really nice, because if the N15 and O18 were showing a very different kind of trend in the upper ocean, it would suggest nitrification. So the addition of nitrate that's been sort of recycled in the upper ocean. And we're not seeing any of that influence here in the eastern equatorial Pacific, although I think we see this down in the Southern Ocean at some in the winter, I think. I don't know what else to show. I have some cool stuff in my office if you want to see. It's a really cool piece of art. And this is a Bruce Springsteen banner. Oh, ow. Dalhousie. What about other sources of iron? From margins, how does that impact your iron input calculation? Yeah, so Al Dalhousie is asking about margin or other sources of iron closer to the margins. Yeah, I'm not going anywhere near the margins. Like, I'm staying very far out in the open ocean, because I know that can get incredibly complicated. Potentially, the measurements that you would see might vary, on much shorter time scales. So you may not get a good picture of the available iron. Yeah. So yeah, and then I try to take variability of dust-borne iron into account in the sensitivity analysis as well. So, for example, one of the sensitivity tests is we just boost the dust input to glacial values, and then there's another sensitivity analysis where we make 60% of the iron available from the dust. It's just and most of these regions aren't very dusty, the eastern equatorial Pacific and the Southern Ocean. So yeah, so it doesn't matter how much you boost dust-borne iron, or how much variability we're not observing from cruises that are looking at dust supply to these regions. But the sediment cores give us a different sense, or at least sort of a longer time scale sense for how much this variability could happen. And so that's why I apply these sort of last glacial maximum sensitivity tests, just to show that even if you, in some cases, I think there's like a 10 times increase in dust during the glacial maximum in some places, and you just can't explain the nitrate consumption. Okay, so we have a question. Oh, I gotta get this one sideways. It's long. Oh, get away. So there's a question about, okay, the first one. Why is the residence time shorter with stronger upwelling? So if you have a fixed depth, say 100 meters, and you have stronger upwelling, you can imagine that though, if you took one water mass, some subsurface source, and brought that through the euphotic zone, that its residence time in the euphotic zone would be shorter. And so when we're at the equatorial Pacific, we're talking about bringing it up to the surface and then diverging it poleward at the surface. So I'm only looking at the equator, so I'm not looking at this stuff that's being laterally input. And so that's where we come up with the residence time. There's a question about changes in carbon sequestration. I'll read it out. Are you assuming changes in carbon sequestration and generally fluxes of elements to depth are constantly in proportion to changes in drawdown, uptake from primary production at the ocean surface? If so, how do you decouple this from changes in export efficiency? Thorium and flux work in the modern ocean both suggest no simple link between drawdown at the surface and export. I totally agree. I'm sort of hitting. I'm I'm framing it like Doug Dale and Gehring and Epley and Peterson did, which is, nitrate consumption. That's the historical way to estimate carbon pump efficiency. I don't, and so that was the motivation, I should say. I didn't actually say anything. I don't think, I hope I shouldn't have said anything about how this model actually impacts carbon export in the ocean, because I agree it's a much more complicated issue. But I think when we are thinking about these things, nitrogen cycling and iron cycling in the upper ocean, that models like this, if they turn out to be true, which I mean, so far I've been trying to disprove it for a couple of years now, that I think we have to consider this, and especially how it may make these regions less sensitive to new iron inputs, like I think about geoengineering. And but in addition, just like you said in your comment, this one comment, it's not clear how at all this changes in nitrate consumption translate into carbon export. So I may in future talks have to make that a little clearer. Have you looked at atmospheric N2O versus nitrate consumption? I've seen some atmospheric N2O records from the ice cores, but a long time ago. There's a lot of high frequency changes, and I'm not sure how. The link I would expect would be via denitrification if we were going to think about ocean nitrogen cycling and atmospheric N2O, which it says atmospheric N2O. So I'm not sure. I'm not 100% certain that we have solid proxy evidence for that at that time scale. Okay, Al, another question. He says he likes the residence time idea. It meshes with a current piece of work. Okay. And he says, let's have a chat about that sometime. Okay, I like that. Okay, another question from Pierce. Would another way to further test the link between upwelling and nitrate consumption be to take the N15 of nitrate measurements at a spot that is further poleward? I see in Piskies that increases in upwelling at the equator, lower N15 locally, it's good, but increase N15 off axis poleward when nitrate is drawn down to completion. Yeah, that makes sense. If we can also have, I guess what you're saying is if we could look at an off-axis, off-equatorial sediment core to kind of reconstruct what this, the changes have occurred between the equator and the off-axis site, then we could pull this apart. It gets a little funky there because you also have to recognize that there's changes in nitrate delta N15 of the source waters. And so that's why in the proxy data I was showing, I actually just call it nitrate consumption because it's actually a corrected value that takes into account the changes in nitrate delta N15. So changes in the delta N15 of the source water. Yeah, it gets a little complicated, but yeah, I think that would work, Pierce. This is fun. I didn't think this would be that fun. It's awkward, for sure. Okay, I'm imagining applause, as Craig says, and okay. Okay, thank you to everyone who came out, and I'll be sending my reimbursements in the mail, right? Thank you.